questions about viruses? Going once, going twice.
Whoa. Hold the horses. The number 15, can you describe the disease path of prion proteins?
Disease path of prion. So, for our prions, there's a lot we don't know about. For those that are infectious prions, the one way we do know you can get it is through digestion. So if you consume prion tissue, basically you can get that prion. What we find is that it's all protein. That means it's only comprised of amino acids, right? There's no virus capsid or anything that's associated with it. You know, what we find is that the prion protein, which we'll just give triangles for its shape, basically changes the three-dimensional shape of a normal part of the protein, which will give circle shapes. So what you end up in the end is with all prion shapes. So for this case, it's a disease about three-dimensional shapes of proteins. So transmission is through digestion or through genetics. You can get it either way. And basically this prion protein changes the shape of a normal protein. It's kind of an amplification process. And they go out, change the shape of other proteins. That would be the best way. Yes?
So, for the first two, the how are viruses classified and what is the viral taxonomy, I pretty much have the exact same answers. Is there a difference?
There is, slightly. So, how are they classified and what was the second part I forgot?
And what is the viral taxonomy?
Viral taxonomy. Of course, I hate taxonomy. Being a virologist, just doesn't work out for viruses. But in this case, we classify based on the type of host, the type of nucleic acid or genome, capsid shape and size, whether it's enveloped, whether it's latent or infectious. There might be one or two other ones in there. We do all this to get what we call the family taxon. Remember, genus species doesn't really work too well with viruses. So with a family taxon, that's when it ends in Viridae. Any other virus questions?
How many examples of the different viruses do you want? It's like, well, I can says, can you give examples of different viruses, AMR, PSS, PSC, and
Right, so for instance, we went through, I was anticipating one of these questions. So pretty much all the ones that we went through. So for instance, now, so basically for animal double-stranded DNA viruses, we talked about the pox, Viridae family. I think we talked about adenoviruses. I can't remember all the RNA viruses we dealt with. Retroviruses, so if you can describe those, HIV, so on and so forth. But pretty much all the ones I talked about in lecture, and I can't remember all the ones I hit on, they would be free game. So you'd want to know their morphology, whether they're enveloped or not, all the stuff we talked about, their genome, of course. I don't know if that helped you out much or not. You'd have to go back to your notes to look. Nope, if I touched upon it, then it's free game. Viruses, again, anybody else? Going once.
Can we go over the growth curve
Viral growth curve.
for replication?
Yeah, and what's the difference between the latent and eclipse?
Eclipse is part of the latent or lag period. So they're kind of one. Of course, I'm going off from memory, so I'm sure I'll mess it up somewhat. So Y virus titer or virus numbers, time on the bottom. Of course, we would have TO. That would be our inoculation or exposure time, however you want to think about that. We get this brief lag, exponential, and then we top off. Whereas a bacterial growth curve is a two-step growth curve. We go up, and then we come back down because it dies. With viruses, we think of this as just a one-step growth curve. We go up, and then they just hang out. We get max burst. So we get lag here, or latent, whatever you want to call that. Exponential, stationary, and this is where we can figure out the burst size. So under that lag or latent, we have that eclipse period, and then all of those things that are associated with the eclipse period, nucleic acid formation, all of that. So when we come out of the latent period, we've gone through all macromolecule synthesis, and we're just starting assembly and release. That's why we're starting to see it go up here at the end. We're actually starting to see release of viruses. Exponential is basically because we have mature viruses and they're just being released ad nauseum. Burst size, the number of viruses per replication round per cell. Does that help you out? Was there another question about this? I felt like it was a two-part question. Oh, well. Viruses again, yes.
What's the difference between virulent and temperate bacteriophage, lytic, and lysogenic?
Cool. So we use these terms kind of synonymously. It has to do whether they're latent or infectious.
Oh, okay.
So if you're an animal, you go through this process, absorption, penetration, uncoating, and it doesn't have to be the exact same number. Depending on who you're talking to, it's like 6 to 8 different things. Then, at this point, we get nucleic acid and other macromolecule synth. They can become infectious, in which case you eventually get assembly and release, or they can be latent. Basically what they do is they take their nucleic acid, and they integrate it into the chromosome of the host. And then they're just there forever until something spurs them on to go back into the infectious cycle.
What's the macro?
Macromolecule synth. Yeah, when I wrote my dissertation for my PhD, I did it all by hand. 178 pages. And then someone had to go back and decipher all my handwriting. I poor wife. I paid her for it though. She was my editor-in-chief.
What was your grade?
Bacteriophage. The same but different. One is the same. And of course, it doesn't get absorbed, so rather than penetration, you get injection of the nucleic acid. You don't get encoding. You do get macromolecule synth. If it goes infectious, with bacteriophage, we call that lytic. If it goes latent in bacteriophage, we call that lysogenic. And same thing, it can hang out for a while, then go into the lytic infectious phase.
So it's just a different name when it's in the bacteriophage versus the animal.
Exactly.
Okay.
And it was actually found in bacteriophage first and then later in eukaryote animal viruses. Gotta be quick. Next, any more viruses going once? Viruses, any other viruses?
Got one more, sorry.
Sure, no problem.
Can you describe the different replication schemes for each genome type?
So I think I can get most of them in my head. So for replication, we also think of this as an expression scheme, because remember, we kind of have to do both in order to assemble, right? So we have to make our macromolecules and we have to express the nucleic acid. And it kind of happens all at the same time. And so think of this as not only expression, but replication kind of together. So it's based upon the genome. If you're a double-stranded DNA virus, everything is straightforward. In order to make proteins and to replicate yourself, it's just like ours. Their DNA gets transcribed into mRNA, translated into protein. If you're a single-stranded DNA virus, you go through a double-stranded DNA intermediate. Transcription to mRNA, translation to protein. If you are, what's the best way to do this? Double-stranded RNA. Your genome consists of one strand that's positive, one strand that's negative. The strand that's positive acts just like mRNA. And it's directly translated to protein. If you're a single-stranded negative, you have to make a positive, so you go through a double-stranded RNA intermediate. That gives you a positive and negative. That positive acts like a mRNA. Is directly translated to protein. Same with the single-stranded positive. I think I'm going to ace this test. I really do. Next one. Viruses anymore? Shoot.
And it makes there's always going to be a positive and a negative in the double-stranded RNA.
Yep.
There's not going to be a virus with just positive or just negative.
Usually not. But that doesn't mean that there isn't. I've learned my lesson not to say never. We know so little that I bet somewhere down the road you would find some that have both. So they would have two positives or two negatives. Not that I know of, but it could be possible.
And the positive and the negative both get translated into protein?
Just the positive.
Just the positive.
Any other viruses before we skip that subject? Boom, microbial growth control. Any questions?
Go over what
Chemotherapeutics.
Yeah.
So we had synthetics and naturals.
Okay.
So the synthetics would be your growth analog, homologs.
That's synthetic.
Synthetic, yeah. Growth biofactors, I can't remember what else is in the synthetic. Quinolones, maybe. Your naturals would be your antibiotics. Any others from the microbial growth control?
Can you describe the different antibiotics and synthetics of their modes of action?
Yes. Can you?
Will you?
I did at one point. I don't know if I could do it from memory. I'd have to go grab that out of my notes.
What do you expect me to
I
memorize?
think I might have that one.
Yeah.
I have that one.
I do indeed.
Harper, I have that one afterwards if you want to look at it.
But I have no, acid is both like,
Quinolone and an antibiotic.
and an antibiotic.
Yep.
Both.
It's a synthetic drug that acts as an antibiotic. How do you use that? But you can use it either way.
Um,
So I wasn't very much help with those. So. I know they're somewhere in your notes. I'm sure of it.
So, do all of the synthetics act like antibiotics?
No, they don't. So for instance, like your growth factor analogs or your nucleotide analogs, they're given in nutritional supplements. So as macromolecules are being made, these analogs get stuck in there. So for instance, if they're nucleotides, they look like a nucleotide. And so when you're replicating RNA and DNA, they get thrown in there, but they gum all the machinery up. They're just not recognized, it all falls apart. Same with your proteins, using other growth factor analogs as well. So like a lot of your proteins need cofactors, coenzymes, vitamins, and if you have molecular analogs that look like a vitamin that's necessary, that aren't, that protein will grab hold of it, then won't be functional. So. Anything else I can't help you with on microbial growth control?
define bacteriostatic and bactericidal.
Bactericidal, anytime you see the suffix "cidal," that means killing
Okay.
or lethal. Static means inhibition.
Kills. Okay.
So a static won't actually kill an organism, but it inhibits it from growing.
That's what you wanted to know.
Those would be the difference between the two, yeah.
Oh, okay. Because in the notes you define the static and cidal, but not bacterial.
So it could be bactericidal, virucidal, fungicidal.
Okay,
Okay.
you just put something there.
Just the cidal at the end
Okay.
means that
That
it's
makes more
killing,
sense.
or
I knew
static
those
means,
two.
right. And then the specific organism we use it towards, then you would just put in the front of there.
Okay.
So whether it's a bacterial, viral, or fungal, or whatever. Any other things on microbial growth control? Going once, twice. You can always email me if you come up with something. Before we move on too much further. Gotta make sure I throw some dates up so we can remember. So don't forget that you have a bunch of stuff due this coming Friday as well, that take-home lab final. All right, next one, bacterial diversity handout. Oh, it sucks, doesn't it?
Never mind, you had it on the virulence one.
What's that?
Never mind, I was on the normal flora and pathogen virulence, I thought we were on something.
Cool.
You could maybe like draw like a tree sort of.
Of all these organisms?
Organize it.
I'm sure there's one in the book or online.
I've seen it, but they're not all,
Cohesive
yeah.
into one tree. Yep.
Some of them are weird, like the Helicobacter is only the endospore forming, isn't that gram-negative?
The Helicobacter, do I have it there? Where's that? Endospore forming and Helicobacter. I think your Helicobacter would be a non-spore forming gram positive. Is that what you were thinking as well? Is it a gram negative? Might be. I don't know why it's in there. I'll take a look at that. But what I would do is know the phylum, what it generally does, which is like the first sentence or two, and then one or two of the examples.
Wait, which, like the very first one, Phylum 1? Or like,
Yeah,
are
that's a
you
good.
talking about knowing?
Most of them only have 3 or 4 at the most. So you would need to, you'd probably need to know 2 out of whatever's there. I won't say which ones. You can memorize whichever ones you think. As an example, so you might see either a matching or a true false that says this phylum has this sort of organism, and this is an organism that represents it, and I give you
Oh, it's like
xylem
those
in
little
this regard.
italicized, names, you need to know those, or like actual bacteria.
For the which?
Just all of them.
For most of them, yeah. So like some of them are pretty easy. Any of the nitrogen are pretty straightforward. So if you have nitrophine or sulfur, a lot of these are pretty straightforward to those genera. You don't see them anywhere else. The methanotropes and methotropes, anytime you see the methyls in front, you know you've got one of those.
These aren't the only phylums, right? They're just the ones we have to learn.
These are the major phylums.
Okay.
Right. Yep.
How many essay questions are there going to be?
Couldn't tell you, haven't written it
Oh.
yet. That way it doesn't get stolen, I found. People don't know what it is, and can't get out there until the very end, so.
Will it most likely be a drawing?
I would say that for your test, you're gonna get lots of true false, maybe some matching, a bunch of short answer and a bunch of essay. Usually for the essay, I give you a choice, say, answer 4 of the 6 or 5 of the 8 or something like that. So you'll be able to choose your strongest ones and try to answer those. Anything else on the phyla? I know lots to memorize there. So, normal flora pathogen, yes.
What are the various types of toxins described? Just like enterotoxins and neurotoxins, it's the only 2 I
So that would be those above, correct? So those would be for exotoxins. We talked about 2 different types, intro and neuro. And then you have your endotoxins. And those would be things like gram-negative, lipopolysaccharides, and some other gram-negative cell wall debris as well. And then those specific exotoxins that are associated with virulence factors. Normal flora, pathogen, virulence.
Go over the first and second one. Yeah, that's what I was
I'm sorry, my ears are bad.
gonna say. The first and second one.
Sure.
11 are there right now.
So first line of defense, think chemical and physical. And these would be in the innate response, right? Non-specific innate. First line of defense. So these are going to be those chemical and physical barriers that we see. Physical, skin, tears, saliva, cilia, etc. Chemical, interferon, high salt concentrations. Lysozyme you find in the tears. Fatty acids, sebaceous stuff you find on the skin, nucleases. Second line of defense. Phagocytosis and inflammation. Does that answer your question? So with phagocytosis, don't forget the Chidiac-Higashi disease, chronic granulomatous syndrome, vice versa. Inflammation, remember it's all about fever, pyrogens, exudative,
Oh, yeah.
purulent,
Oh, I was gonna ask you, what is oxidative burst?
oxidative burst. So during phagocytosis and inflammation, that white blood cell, whether it be a macrophage, probably more of a nuclear, whatever it might be, ends up phagocytizing that pathogen into that phagolysosome, or lysosome as we call it. And because it's in this internal environment, this is a membrane that goes around it, it can actually manipulate the environment within that phagolysosome without manipulating it everywhere else. You get an increase in proteinases, a decrease in pH, and oxidative burst. And basically what that is O2 metabolism of glycogen stores within that phagolysosome specifically. Glycogen just being polymer chains of glucose goes straight into glycolysis. Into Krebs, we get some really bad byproducts. Super free radicals. O2 is a negative charge. Hydroxide free radicals. H2O2 being produced. All of those are really detrimental, lethal to the pathogen. All of this together basically chops that pathogen up into bits. So it's the way that the pathogen gets degraded down into those antigenic bits.
What's interferon?
Interferon. So interferon is a chemical that is produced by virus-infected cells. So if I had my line of tissue cells here, and all of a sudden one of these gets infected by a virus, that virus-infected cell produces this chemical called interferon, IFN is what we'll call it. IFN is absorbed by the neighboring cells, and the neighboring cells make all of these antivirals. Anti-protein, anti-nucleic acid, they form pore holes in that viral cell. And even though it's non-specific, it still kind of gets modulated a little bit with virus infection. It's not turned on until your very first viral infection after you've been born, and then from that point on, because you're bumping into a million viruses every day, it's just kind of always turned on. Next question. Anything else on normal flora, pathogen, biovirulence? Going once, going twice? Excellent. Last few left, immune responses. Go for it.
Can you sketch the molecular structure of antigen?
Yes, can you?
Not quite.
So remember, 2 heavy chains, 2 light chains. also called too long, too short, depending on how different they are. Remember, they just form a Y with the heavy chains on the inside, light chains on the outside, held together by disulfide bonds. You have the FC region at one side, the Fab or antigen binding regions at the other side. Shoot them at me. Any more?
What cells are involved in the adaptive response?
So, two different types of adaptive responses, remember? Humoral and cellular. Humoral starts out with a white blood cell. It goes through phagocytosis, like we just talked about. Ends up being an antigen-presenting cell. Via MHC2, it interacts with the T helper. That produces a bunch of cytokines, helps with B cell differentiation into a plasma cell. And that plasma cell eventually gives us antibody at the end. For your cellular, that's just any sort of virus-infected tissue or tumor tissue. It then becomes an antigen-presenting cell via MHC class 1, interacts with T cytotoxic, which get differentiated into cytotoxic T lymphocytes. That, along with natural killer cells and other recruited cells, deliver a lethal hit back to the virus tissue. Does that help?
Are we going to need to draw the picture that you drew originally?
Yep, you should be able to sketch both of these completely. Tell me what the beginning products are, end products, what happens, what it does, and specifically that MHC TCR T cell interaction we've talked about.
Do you want the picture or not?
I would say the question would probably say sketch and describe. So I would probably want both.
Okay, I have another question.
Shoot.
What is PMMS mean? So like the leukopenia and the leukocytosis?
Leukopenia and leukocytosis,
Does
yeah.
this increase in PMMS and decrease, what is that?
Polymorphonuclears, so the PMNs.
Oh,
Those are type of white blood cells. PMN and then the little s after it. So increase or decrease in these white blood cells with infection, which is strange because most bacteria give us an increase in white blood cells, not all the time. Some gram negatives can make a decrease. Many times viruses will decrease our white blood cell counts. It can go both ways. Any other questions? Going once, going twice. Immune responses, we're good there. Everyone's good. Antibodies, classes. Antiserum and vaccines.
when are they normally used?
Which ones?
Is it, oh, antiserum, sorry.
Antiserums. So remember, this is basically someone's serum, right? With all those polyclonal antibodies. So we use them in a few different ways. Prophylactically, we call this IG therapy. So it just gives us very short what would we say, protection or coverage, usually within 2 weeks. So it gives us immediate short protection. So we can use it as a prophylactic if you bumped into someone with hepatitis C or whatever. We can give you IG therapy for that. We can also give you IG therapy for specific pathogens. So if you have some type of salmonella type that we have, and then of course with toxins, we can basically use those as antitoxins, so or neutralizing antibodies. Those would be examples of when we would use them.
What are the pros and cons of protein vaccines as compared to DNA vaccines?
So protein vaccines, pros and cons. So proteins are pretty expensive to develop and to dispense. Short shelf life. Got to make sure you take care of them. Heat labile, meaning they're pretty susceptible to any sort of heat. Multiple administrations. So usually you need some sort of booster. And of course we can have different side reactions, specifically things like an allergic reaction. The pros of all of this many of the methods are already developed for doing all of this, so once we have an assembly line for making protein vaccines, we can very quickly shift it from one protein to another. And so these are all pretty much up and developed by most of your vaccine makers. And they work really well. So they're good and efficient. DNA. They're cheap to research develop, cheap to administer, super long shelf life because they're basically just dehydrated nucleic acids. They are heat stable when they're dehydrated like that, so you can keep them as long as you don't burn them in the oven. There's no real temperature, natural temperature out there that burn them too bad. Usually we get one single administration. And whereas this is usually towards few antigens, this can be towards many different antigens from many different organisms. And that's pretty good there. Cons, of course. We don't know a lot about them. Do we end up getting recombination with host cells or with other pathogens? Can we get an antibody response toward these foreign, I forgot how to spell foreign, DNAs? I was close to foreign. Non-self. And there have been some clinical problems. So they haven't quite made it out of the clinical setting yet. And that's about all I have for that one. Any other vaccines or anti-serum questions?
Yes. What do we consider when contemplating the efficacy of a vaccine?
Of the vaccine. So, if I remember rightly, let me put this together in my head. Oh, God. It's right there, hold on a second. So efficacy, how well it works, compared to side effects, I believe.
Is it administered properly, stored properly?
Administration,
Contamination.
storage, I believe.
Contamination
And can't remember which other ones were there. Any other vaccine or therapy ones?
What is an ideal vaccine?
An ideal vaccine, so that would be one that's cheap, easy to research and develop, long shelf life, one administration, no side effects. Just think about everything vaccines are. And so if they shoot for these, or as many of these as possible, then they get the best vaccine that they can. And I think I got most of them. Any others, vaccines, anti-serums, going once, going twice? Healthcare epidemiology, any questions on that one?
Yeah, are we just doing literally almost everything from the books?
Pretty much so. Know those general terms, morbidity, mortality, prevalence, point prevalence.
All the percentages.
Yeah, exactly, for all the healthcare associated infections, know which ones cause what, the amount of death you get from each of those. All right, so for those of you that are showing up on Wednesday, 6 o'clock, you'll have as much time as you want. If you're not showing up on Wednesday, you need to show up on Monday, same time, same place. Any other questions? I'll hope to have most of your lab stuff completed, if not complete by then. I have a pretty rough estimate of your grade now, but I'd have a full one on Wednesday. So if you want to show up and grab it on Wednesday, whether you're taking the test or not,
Just come here.
I can just give it to you. Any other questions? Go for it. If you need any of this stuff, it's up here. Review material, supplemental notes,